Kia ora. I'm Associate Professor Diane Kenwright from the University of Otago, Wellington. I'm going to explain to you some cervical histopathology, which is important in understanding cervical cytology. The cervix has an ectocervix and an endocervix, and between those there is the transformation zone. The ectocervix is covered by squamous epithelium, and the endocervix is covered by glandular epithelium folded into crypts. We're going to think about now the transformation zone because this is where the most significant HPV effects occur. Before puberty, the transition between endocervix and ectocervix occurs here inside the endocervical canal. As puberty occurs, the glandular epithelium moves outside onto the outer facing surface of the cervix. And then, as the woman ages, this epithelium transforms back into squamous epithelium so that in a woman's 30s, there is now squamous epithelium again over this surface. This area, which was originally glandular and then recovered by squamous epithelium, is called the transformation zone. Histologically, the ectocervix is covered by glycogenated, non-keratinizing squamous epithelium. If we look at the squamous epithelium, it's got a regular line of small basal cells and then a couple of layers of parabasal cells, which then mature into glycogenated squamous epithelium. The endocervix and its crypts and glands are lined by glandular epithelium, which is characterized by a single layer of cells with basal nuclei and then apical mucin. Here we have the transformation zone. There are a couple of things to observe. The first thing to observe is that there is squamous epithelium at the top where it has grown over the glandular epithelium. And the second thing to observe is that all these glands are connected to the surface and are really crypts, not true glands. Let's take a closer look. Here we can see the normal squamous epithelium with its basal and parabasal cells overlying these crypts that we will call glands from now on, lined by a single layer of columnar cells with apical mucin. Sometimes the normal squamous epithelium extends down into these endocervical glands. This is called squamous metaplasia because the glandular epithelium is turning into the squamous epithelium and is entirely normal and physiological. This is important because later we're going to be looking at squamous cell carcinoma. Just because the squamous epithelium is inside glands, this does not imply dysplasia or malignancy. It is a normal process. HPV infection can result in a variety of changes to the squamous epithelium. The first one shown here is what we call HPV cytopathic effect. This is when the HPV has infected the cell and remains inside the cytoplasm of the cell. It causes the cells to enlarge and often become multinucleated. You can see that these cells are slightly darker. This is nuclear hyperchromasia, and the nuclear membranes are irregular. Some of the cells, such as these ones here, have clear halos around them. This is what characterizes a koilocyte and is the pathognomic change of HPV infection. After HPV infection, we can get some atypia in the epithelium without it being integrated into the nucleus. This is what causes CIN1. Here, looking at the base, we can see that regular row of basal cells, like a picket fence, has now gone, and these cells are now more irregular. The basal area has been expanded, and the cells themselves are larger than normal. Here we can see really prominent koilocytes with their enlarged hyperchromatic irregular nucleus and their perinuclear halos. High-risk HPV types have the ability to integrate into the cellular DNA, and viral oncogenes are activated. In this picture, we can see an area of CIN1 similar to that which we've previously looked at, and an area of CIN2. CIN2 is classified as a high-grade squamous intraepithelial lesion. You can see the cells are larger and more pleomorphic. You can see that the thickness of the atypical cells extends to the midpoint of the epithelium. You can see that there are atypical mitoses. The koilocytes that are seen in CIN1 are also seen in CIN2. One thing to note here is that both changes have abnormalities right to the top. It's just that it's the koilocytes extending to the top, not the basal cells. It's the thickness of the basal cells that you will find very useful in determining the difference between CIN1 and CIN2. In this photograph of CIN2, notice the basal cells extend to halfway, anything between 1/3 and 2/3 of the thickness of the epithelium containing atypical immature basaloid type cells defines CIN2. We have multinucleate cells. We have mitoses occurring in the mid 1/3 of the epithelium. This might be one here. And we have irregular hyperchromatic nuclei in basaloid cells confined below the 2/3 mark, and then koilocytes here at the top. In CIN3, we have these immature basaloid cells with a high nuclear cytoplasmic ratio now occupying the full thickness of the epithelium, extending into the upper third of the epithelium, and they're accompanied by mitoses extending into the upper third of the epithelium. Abnormal mitotic figures and irregular hyperchromatic nuclei remain a feature of CIN3. There are benign changes that can mimic CIN. They do this by causing enlargement of nuclei and hyperchromasia. Here, for example, inflammation has caused these nuclei to enlarge, develop small nucleoli, and this immature area has got these cells extending to the top. This doesn't mean that it is CIN because they lack the irregular nuclear outline, overlapping, and dense packing and hyperchromasia of CIN3. But the lesson here is that not all changes that have these basal type cells extending to the surface are CIN. Cytology is particularly useful in telling the difference between these conditions. Let's have a look now at squamous cell carcinoma. Squamous cell carcinoma arises from high-grade squamous intraepithelial lesions. So here we have CIN3. It has extended down into glands like the squamous metaplasia, but in this area here it's still bounded by the basement membrane and so is not invasive. Just here we can see the earliest sign of invasive squamous cell carcinoma. We have a small bud of cells that's breaking through the basement membrane and extending into the underlying stroma. They look nearly the same as the CIN3 cells. Around it are a whole lot of lymphocytes and inflammation. Here is another example of very early squamous cell carcinoma. We can see the CIN3 with the intact basement membrane coming around here, and then a couple of buds breaking through the basement membrane, and here another bud breaking through the basement membrane. Look what it's doing. It's becoming pinker. This is called paradoxical differentiation. And in cytology, you will be able to see cells in squamous cell carcinoma that show this paradoxical differentiation. They start to look like tadpoles and elongated cells, and some of them have quite pink, copious cytoplasm. This change can indicate that there is early squamous cell carcinoma invading the underlying stroma. This is the next stage in invasive squamous cell carcinoma, where the little buds are now breaking off and cells are migrating down into the underlying soft tissue of the cervix. As squamous cell carcinomas develop into larger tumors, they can develop these nests in which there are keratinizing cells. And we call these invasive squamous cell carcinomas with a keratinizing morphology. Sometimes the cells don't mature in their little nests and form keratin, and they remain this dark blue color resembling CIN3. This is a non-keratinizing squamous cell carcinoma. Both these squamous cell carcinomas will be related to HPV. Very few squamous cell carcinomas are not related to HPV. Finally, my key message. In order to be a great diagnostician, you must use your histology knowledge and cytology knowledge together to make an accurate diagnosis. The observational skills you will learn in cytology will help your histology immensely. Thank you.